I'm Art Motozenik, and I'm with the American Association of Pharmaceutical Scientists. It's my pleasure to host a series of interviews with distinguished pharmaceutical scientists who have made a major contribution to our profession in pharmaceutical research. Speaking on behalf of the AAPS Distance Learning Committee, it is our intent to try to capture the inspiration, the career path, and perhaps the major contributions of distinguished pharmaceutical scientists and share this with the academic, industrial, and research communities, as well as industrial scientists in pharmaceutical sciences. We'd like to explore the kind of career path decisions that they made in shaping their directions in their career in research. We'd also want to explore the perspectives that they might have on the future of pharmaceutical research. Today it's my pleasure to have an opportunity to visit with Dr. Vincent H.L. Vince is a friend and a personal colleague that I've known for many years. He has had a very distinguished academic career at the University of Southern California School of Pharmacy, where he was a distinguished professor. And currently he's the Associate Director of the Office of Pharmaceutical Science at the Food and Drug Administration, a big transition in Vince's career. So Vince, look forward to hearing some of your comments. I'd like to ask you to begin, however, if you could track for us some of the early formal training that brought you into pharmaceutical sciences as a career path, maybe beginning at the university level, and tell us what was it that made you make those decisions to seek the fields that you entered.
Well, Art, it is a pleasure to be here. And first of all, I am a scientist by accident. I aspired to be an artist early on in my career. And then, but my late father persuaded me not to do so, because he said unless I knew that I would be an outstanding artist, I should not pursue it. My entry into into science was inspired by two or three professors who took an active interest in my career. To name one of them was Professor Nienhuis at Fair State University. And he was an organic chemist, and I have a passion for organic chemistry. But at the time of my decision to go to graduate school, there was no jobs for organic chemists. And moreover, that I set a laboratory on fire while doing synthesis.
You actually set a laboratory
on fire? Yes, that's right. So he persuaded me not to pursue organic chemistry. The second person who had an impact on my career was the late Professor Michelson at Fair State. He persuaded me not to pursue medicine as a career. He said that medicine is a very boring profession, and I believe in him. So, but he was the one who was instrumental to get me to the University of Wisconsin, the best school of all times, as you know. And there I met Professor Joseph Robinson, who has a profound effect on my personal and professional life. Joe taught me how to succeed. He opened doors for me. And to this day, that I practice what I learned from him. And also, he was very generous in mentioning my name whenever he was approached for work in alcoholic drug delivery. And Joe was very generous in sharing that field with me. And then one more person who has a profound effect on my career was my former dean, John Biles, who was very persistent in pursuing me. As you know, that you and I were talking some time about pursuing a different career. But it was John who gave me the confidence to pursue it. And on this note, there's also one more person I would like to acknowledge, was Dr. Jordan Cohen, who was my first department chair. And to this day, that he was still interested in me as a person. And as
but Jordan Cohen, of course, was by that time at the University of Southern California.
That's right.
Correct? Yes.
Yep. And he looked after me. And so he has a profound impact on the way that I conduct my responsibility as a chair of the department.
As you completed your initial training at the University of Wisconsin, you were motivated to go into academic research immediately, or did you consider industry or other options?
Well, Art, I did consider industrial research as an option. And amazingly to me, that even though I have no training in industry, that I had the confidence of the scientists like yourself in me, but in my heart, that I wanted to be in academic research. I don't know why, but I believe at that time it was because I love teaching. I love to interact with younger people, and also even though I was persuaded not to pursue it, not because of I don't know for what reason, but primarily because of the competition in funding. That was in 1979. Getting funding from NIH was extremely competitive, but I want to give myself a chance, and also it was the former dean John Biles who gave me the confidence to pursue it.
Right. As you entered the academic ranks, you continued some of the early work that you had done in ophthalmic drug delivery and pursued that to a point that's one of the hallmarks of your career. You've been a major contributor with some seminal work in that area. How did you, for how long did you continue that interest in ophthalmic research and drug delivery and barriers to ophthalmic drug delivery? And then when did you branch out into other areas?
Well, that's a very interesting question, Art. To this day, I still have a passion for ophthalmic drug delivery. In fact, even at the FDA, when I do not have a research lab, I still follow the field. And I will not be surprised that someday I might go back to pursue what I wanted to do. I also understand and realize that to be successful, that it would be unwise, let me put it this way, to compete directly with Joe as a scientific colleague. Joe was very generous in sharing the field with me. But even though he was very generous, I realized that given his extensive experience, that I would be silly to compete that on. And that brought me to the next field is that I have to do something differently. Now, the eye was a very small organ, but there's plenty of work to be done. So instead of pursuing the cornea, which was Joe's major contribution area, that I pursued the conjunctiva, which is the tissue next to it. And one thing led to another, that in fact that my group, former group, was the only group in the world to pursue that. The area I chose was not just by accident, but with planning, something that I would like to share with the next generation. That I have, in my pursuing my graduate research, that I already have decided what to pursue, and that is peptide and protein drug delivery. Joe assigned me on a very risky project for which there's no background in the lab. I had to contact scientists who are expert in the area, in biochemistry, molecular biology, and so on and so forth. And at that time I said that, well, he wanted me to characterize a substance called mucin, to design mucoadhesive polymers. And so I decided that instead of using proteins as scaffolds, I look at protein and peptide drugs.
And that brought you into your extensive work in protein and peptide drug delivery and the barriers of drug. Right. Yes. And so you're indicating to us that you were very interested in new multidiscipline approaches, certainly physiology, looking at organ transports and so forth. Did you find a necessity to branch out and reach across disciplines in the medical community, clinical communities, as looking for collaborators and so forth?
Yes, I certainly believe so. And in fact, Joe was instrumental in broadening the horizons of his students. He constantly preached, and I use that word very deliberately, to us that we need to know areas outside of our own field. Pharmaceutical science is an applied area, and it's very essential for us to know what's going on and bring the new advances in other fields into this discipline, the pharmaceutical sciences. And also, I realized that to be successful, I need to learn from role models. And I can name many role models, and one of my hobbies is to understand why people are successful and what does it take to be a leader.
Well, you have a very deserved reputation for infatigable energy and applying a lot of hard work into a new field, and you plow into it, explore it, and then you take it forward. Do you have any areas that you have a high degree of satisfaction in feeling retrospectively as you look at how you open up new categories of research that you might want to share with us?
Yes, I think this is an excellent question, Art. In retrospect, if there's one common there's one feature that I feel proud of is the observation that Mother Nature designed us in a very simple way. And there's always a plan A and plan B. And the plan B that I pursued was the conjunctiva. The conjunctiva is an area that was identified by Joe's work as an area of drug loss, rightly so because of this blood circulation at that time. And I was intriguing in that, so that it was believed a long time that it's impossible for drug in eye drops to get to the back of the eye. In fact, in the 1990s, extensive effort was devoted to keep drugs away from the back of the eye. But now in the 21st century, we want to get drugs to the back of the eye. How can we design molecules to match the barrier? And this is an area that I've been pushing constantly, even to this day.
Well, that's a very interesting observation you made, because you certainly changed the course of pharmaceutical research by focusing on the different areas of drug transport there. Would you comment about how you were interested in looking at structure relationships and structure-activity relationships and elucidating those mechanisms for drug transport, because you've been a major contributor in that area?
Well, thank you, Art. That brings me to the academic environment that one is in. And oftentimes, I also believe that we need to have a target. And here I would like to mention that I learned from Gordon Eminent, a distinguished scientist.
Well, I think that your collaborations with other eminent scientists are well known. I would also suggest that you have been a very significant contributor to the collaborative researches that comes from publication, etc. You have probably one of the most distinguished careers of service as an editor. Mm-hmm. And I was wondering, did you find that that was an addition to your work or a distraction? And how did you come to be so collaborative in so many other research contributors with the literature field, as an editor of pharmaceutical research, for example, and your contributions to advanced drug delivery as an editor as well?
Well, this is something that I see as my contributions, my obligations to the field. I got into editing in part through my experience in Joe's laboratory. That he was an editor himself, so I learned the mechanics from him. But also, I see that it's very important for the editor to shape the course of research in a field. And I take this responsibility very seriously because many of these authors are colleagues, are friends. But I would like to see my role not as an auditor or judge, but as a mentor, as one of the participants in the scientific community. My operating philosophy is that I need to identify the area that is interesting, that's important. I like to highlight it. I want to talk about it. I like to engage other scientists to follow that path. And I think this is a responsibility that is essential.
If we examine your career in the sense of the early career, the mid-career, and your current activities, would you look backward retrospectively in your early and middle career and find that there was a time when you made a pivotal decision to focus in one area and specialize in it? And what made you come to those decisions?
Well, that was, again, going back to Eminent's operating philosophy, is that I learned from Gordon that everything is a moving target, but a moving target is better than no target. And I think as most of the work that we pursue, that we should liken it to hiking. You go by your instinct, you take a look at the landscape, and see what might be a path to take. And that's what has been guiding my career.
Who did you feel were contributors to changing a direction, maybe making a pivotal choice in your career path, because you had lots of options?
Yeah. Oh, there's a colleague at USC that I was determined to understand under what conditions can I get drugs to the back of the eye.
And who was that colleague?
There was Dr. Alicia McDonald.
Right.
She was a member on the PhD committee of a student, Uday Kampela, a former student, now a colleague in pharmaceutical sciences. She observation is that for us to be successful in drug delivery, that we really need to understand the cell biology and the physiology of the tissue that we work on. And that brought me into contact with a collaborator, Dr. Kwan Jin Kim. And Kwan is an engineer, is a physiologist, and he looked at our work from a different perspective. And that reminded me of another important element to the fun that I have in my career, is to learn from him. And I firmly believe that collaboration is essential, as you know, especially in today's climate. We need to know not only our colleague from a scientific level, but as from friendship. I think there's also another element that contributes to success. So that was a turning point. But one of the final thing I would like to say is that that's why the advisor needs to be firm. Students oftentimes like to take the low energy path, as we all do. But I see myself as a coach. That my job is to recognize the potential of my students, raise the bar as high as possible, but not to break the back of the athlete.
Well, that's well said. Did that give you a great degree of satisfaction to see people who are influenced perhaps by your mentoring and moving in those areas?
Certainly, Arch. Also, the other thing I recognize is that I cannot change you or anybody else, because by the time you come to my laboratory, shall we say, that you have been shaped in the formative years. And the only thing I can do is to learn about what turns you on and how will you fit into the team. And I think this is the most frightening part of my job, shall we say, as a professor, as a supervisor, is to how can we know from a day or two interview that this person would be a good fit, and how can I convince the rest of the team that I handled this team member differently than them.
We've been professional colleagues for many years, and we've known each other very well. And I recall that during the era of the 80s, perhaps, when drug delivery was emerging as a major discipline, you were one of the early leaders in looking at control release requirements and looking at the requirements addressing the physiological barriers to drug release. Was that a very pivotal period in your career, and then did you choose to pursue that on a collaborative basis, or were you pretty much a singular leader in some of these areas?
Well, I don't believe that I know sometimes I thought I knew everything, but in truth that we don't. And what we need to do is identify how to ask questions. This is something that the scientist or the scientific team leader has to do. And then look at who else would be important contributors to the success of this work. And this philosophy has been guiding my career.
And at that time, did you feel that you had any people that were important and singular in your motivation, maybe even on a personal level in your family, or some of the other people who were inspiring you to continue in your work at that time?
Well, certainly that the experience I have in Joe's laboratory was pivotal. Joe is a leader in controlled drug delivery. And as a student, he asked me to work with him on the book called Controlled Drug Delivery. It was through reading these manuscripts from page 1 to page 2, page 1 to the end of the book, that I got to learn the development in the field. I got to learn the major scientists in the field. And from that perspective, I know what are some of the gaps in the field. And that sustained my interest. And also, at that time, that I was not quite sure about how can we define drug delivery. But also, I believe at that time, that it was essential to be collaborative.
Part of the challenge, of course, in drug delivery is to be able to be as quantitative as possible and to translate those quantitative measurements into significant factors in, perhaps drug efficacy or safety, which brings us to the regulatory environment. Yep. Now, you now have an active career at the present time in the regulatory agency. When did you first get interested in, let's say, regulatory hurdles to demonstrate proof and evidence of safety and efficacy?
Well, this is an aspect that I never planned in my career, Art. But I do have a plan to contribute to the public policy, but not at this stage of my career. But when the opportunity presented itself, that I said, why not? And you mentioned the word quantitative, and that reminded me about what I learned in graduate school. Is that I was told that unless you can quantify an observation, that's not science, and that was mentioned by Takiguchi. Paraphrasing Takiguchi, a giant of our field. And it's also interesting to see how science would come around in full circle. But in direct answer to your questions, the quantitation is something that we need to insist on. And along the lines of quantitations, that I would like to emphasize that we need to insist on mechanisms, understanding the mechanisms of why something works, why something that doesn't work. I think that, not to be criticism about the industry, is that the pressure to bring a product to the market oftentimes is at the expense of understanding science. So I hoped that the regulatory agency would set a new standard to elevate the level of science in the development of products. Not that science is not guiding it, but I think that we can do much better.
You also have an acknowledged reputation for assisting people from many other countries with the international connections you've had. How did you get involved in the collaboration and the teaching and the sharing of ideas and the leadership role you've had in global pharmaceutical science?
Well, Art, that I am a traveler, and I love meeting other people. And I think that is a dimension that must be emphasized in today's world. As you know, the world is flat, as a book by Freeman indicated.
Right. And
the world is flat. It depends on how far you look ahead. I think that the culture element is essential. This is an element that we must take advantage of in the laboratory. I'm an international student. I was an international student, and I was very sensitive to assimilate myself into the American culture. But at the same time, I think that we have a lot to learn from the other cultures. As you know, that research is now not only multidisciplinary, but multicultural. And the way that we conduct our business, whatever business that is strongly influenced by our heritage. And I think that we need to understand that in interacting with our colleagues. And we need to respect their heritage in the pursuit of our common goal.
I'd be remiss if I didn't acknowledge the fact that you have served as president of the AAPS, which is sponsoring this series of distinguished lectures. During the period of time that you were president, you forged a very strong leadership role in moving the association into new directions. Could you share with us some of the feelings you have that you're most proud of, or you felt that the group and the team that you assembled and worked with at that time made a contribution? Where would you see that as being pivotal?
Well, there are two or three things, Art. The first thing is to look at what has been accomplished in the past. Predecessors like yourself, that I did have an agenda, but I think it would be not be valuable to the association if I have to start from scratch. The association is strong because of the foundation laid by previous colleagues. At that time, that I also realized that I only have 1 year to accomplish what I wanted to do. So about 12 months before my presidency was to begin, I consulted with a number of colleagues and said what would be best for the association, and I identified 2 or 3 areas. One is that how can we make sure that we have the diversity in science and in countries in our future membership. I think that we always should look at the future. Where would that be 5 to 10 years from now? And we identified the students as one area. And today, as you know, that we have in excess of 40 student chapters from all over the world. That's very satisfying. The other element I noticed is that we cannot, the second element is global alliance. In the sense that at that time, in 1996, the science is clearly becoming globalized. I don't think any country can claim monopoly of science. And I think that in the immediate future, that we need to know where the research excellence is and forge an alliance with that region.
Right.
And the third thing related to this is how can we have an effective organizational structure that allow decision making to occur in a expedient process, and yet would not give the membership the impression that their opinions was not valued. And so I decided to restructure the organization, and that was a very interesting task, because it would come across as being threatening. And one last thing that I would like to mention is that it takes skills to work with leader colleagues on a team whose position on the team was not because of me, because they were elected independently, and they also want to contribute. So to identify what turned them on, what do they see as important, and be able to convince them this is the difficult but wise things to do, is very gratifying. I would like to mention Alice Till and Richard Bergman, the two colleagues who were instrumental to provide me the guidance about bringing about change.
Well, you're very generous in acknowledging and sharing recognition with your colleagues, and it's well deserved. I'd like you to, we've reflected on your mid-career and your current activities. I wonder if we could ask you, what do you think are the biggest challenges in pharmaceutical research today? And then I'd like you to follow up by suggesting where you see in the short-term future some of the challenges emerging.
One of the important challenges in pharmaceutical sciences today is to recognize that we are the best. We are very good. And we need to demonstrate that by reaching out into the supportive science, or the basic science. It's in my observation, Arch, that we might have a very modest approach to our accomplishments. We need to strengthen our outreach efforts, and this is what the AAPS and other organizations can do, is to promote the accomplishment of pharmaceutical scientists. And I think that can only be successful if we have the substance to show it. Mm-hmm. We have to profile distinguished scientists. I'm not sure whether I belong to that group, but we need to have role models to inspire the next generation. Mm-hmm. We need to get into the habit of promoting our colleagues in being reviewers, in being members of grant review panels, to promote pharmaceutical sciences. Sometimes I feel that we are overcritical of ourselves, mm-hmm. But we need to be the advocate. So if we are elected to a prestigious body, it's our responsibility to promote colleagues who are qualified. And this is an area for which I think that we need lots of work. We need to share this kind of an obligation to a colleague. That pharmaceutical science is a very essential science to bring a product about, especially in today's world of translational research.
Well, I think that's very, very well said, because it appreciates the fact that collaborative science is important, even challenging science where there is critique and overview. And you've certainly promoted that through your leadership positions in the journals and editorial reviews and so forth. Do you feel young scientists today who are entering the profession, what advice would you offer them in terms of what they might look to, based on your own experiences, but also on what you would consider a charting for the future?
Well, Art, this is an excellent point. My advice to the students at this point in time, based on my observation in various capacities, is that the younger generation is somewhat impatient of being recognized. That I think they ought to spend time to solidify their science than to be distracted into other activities. I think that's essential. I think they need to feel comfortable to reach out into other sciences. And that's just like going to a different country, is to learn what their culture is. I think the best way to learn a language is to learn about the culture. So we need to learn about the culture of our engineering scientists, colleagues, and physicians. I think that we need to, from that perspective, that we need to, from the professor point of view, to give opportunities to improve the communication skills of our students. I think, as you know, this is a skill that we must have to, in industry, academia, regulatory agencies, or wherever you practice, the ability to convince your colleagues that they're not working for you, but working for a common goal, and share the excitement. But one thing I would like to mention is that when we engage in a project, let's do it with the final goal in mind, rather than who is going to be the first or the last author.
In terms of the accomplishments of your own work and the team and your students and everything else, where do you see your important legacy? What are you most proud of in terms of the contributions of Vincent Lee and his students and his collaborators and so forth?
Well, that's a maybe premature question to ask, Art.
I'm thinking about your satisfaction.
Oh. Well, the most satisfying is to see my colleagues succeed. And I think this is an area for which I'm not blowing my own horns, but we should take pride in the success of our colleagues, rather than being jealous of it. And I got to this stage in large part because of the outstanding scientists around me, and I actively pursue them. Also, I'd like to share one observation. Les Bennett. I used to think that Les was not approachable. I also thought that Gary Levy, the giants in the field. But when it comes down to it, we're all human beings. We all will act differently on stage. But when it comes down to it, that we are really have the future of the science, of the field at heart. So I would encourage the junior colleagues not to be intimidated by approaching distinguished scientists.
Well, I think that the point you make about outreach and communicating and sharing ideas, even disagreeing but being candid and open, is very important to fostering good synergized science. And I think that's a very interesting observation. Do you feel that there are barriers to that kind of communication that young people have to try to overcome?
I think the barriers, if I see it, is the confidence to pursue the goal. And I think that we have to overcome that barrier. And that's not going to be happening overnight. It takes time. And I think that brings me to the final point, an important point I'd like to mention. No matter who we are, as a graduate advisor, as the supervisor of a scientist in industry or regulatory agencies, that we need to put the career and the professional growth of our colleague in mind. And this is something that in my observations, in my different aspects of my career, that perhaps that not all of us have put that aspect as a first priority.
Based on the influences in your life and your career path, might you comment on your personal relationships, perhaps with your family? Were they influential in some of the things that you...
Well, it's that we all have families, and I don't think the family got enough credit. Sometimes we take things for granted, but it ought not to be. I'd like to say that my family, my wife, and my children, my 3 children.
And your wife's name?
Elaine. And my 3 children are Amanda. She is an artist, and I hope that and also a story writer. That I hope that she would be writing the next Harry Potter. Ryan is 15 years old, and he wants to go to Duke and pursue a business career. And Justin, 11 years old, and he is the gifted child.
Wonderful. You know.
And I think that he inherited the gene from his mother.
Well, you sound like a very proud husband and father, and I congratulate you on your family life and the inspiration they've had for you and your career path, and your wife certainly as your intellectual and family partner.
Thank you.
Well, I certainly want to congratulate you on a very active career, which is still very much in motion, and applaud your interest in helping the young scientists and offering advice. So Vince, it's been a pleasure interviewing you.
Thank you.
And I wish you well in your efforts at the Food and Drug Administration. I certainly look forward to the kind of opportunities that you're going to present to the scientists at the agency as well. Thank you.
Thank you very much, Howard.